Thank you, Rebecca, and thank you all so much for coming tonight. I'm really excited to introduce this topic to you. So I want to start out by asking you all a question. How many of you have been to areas where Zika, chikungunya, or dengue are being transmitted? Show of hands. Okay. Just let me hear some of the places you've been. Jim, where have you been?
Parts of Central Africa.
Okay. And over here, where have you been? Nicaragua. Okay. Where else? People? Cuba, India?
Brazil.
Yes.
Key West.
Key West. Absolutely. Okay. So that leads me into talking about that we don't only see these diseases being transmitted outside of the U.S., but also inside of the U.S. And dengue is the first one I'm going to talk about. You can see here that in 2010, Florida confirmed 24 cases of dengue in Key West. We have dengue in Texas, and we have dengue in Hawaii. We have actually 400 million infections around the world annually, and just under a quarter of those manifest as apparent disease. But we also have chikungunya, another mosquito-borne virus that swept through the Caribbean starting in 2014, rapidly spread throughout the Americas, and to date we've seen over 1 million cases in the Americas alone. And the third one is Zika, one that I'm sure you've heard a lot about last year. This exploded in Brazil starting in 2015. Also, over 1 million cases in the Americas. The World Health Organization declared Zika a public health emergency in February of last year. We've seen transmission in Florida, as well as Texas in the continental United States. So where have these recent outbreaks of Aedes aegypti-borne viruses occurred in the continental United States? You can see from the key that the blue is dengue, the orangish is chikungunya, and the purple is Zika. So I've only gone back as far as 2010, though we've seen dengue sporadically in Texas since the 1980s. We used to have a lot of these diseases in the US before the advent of DDT. We started spraying DDT, we saw a complete reduction in mosquitoes and diseases, and now gradually we're starting to see things change. So 2010, we had 65 cases in Key West, Florida, and then you can see throughout the years that we've had dengue cases in Florida and Texas, and then in 2014, chikungunya emerged in Florida. 2015, we only saw 1 case of chikungunya in Texas and 1 case of dengue, but then look at 2016 with Zika. 256 cases of Zika in Florida, 6 in Texas, and just 2 of dengue. But what this map is showing you is locally, cases that are locally acquired. These are not cases from people who've traveled to Cuba or Nicaragua or India and brought the disease back to the US. These are people who acquired these viruses in the United States. And we know that because whenever somebody comes in for testing, we always do travel histories on them to see where they've been. So I want to talk just a little bit about these 3 viruses and compare the 3 of them. So the first one is dengue. It's often less than affectionately referred to as breakbone fever because it feels as if every bone in your body is breaking. It has a severe hemorrhagic form. There are 4 serotypes of this. And when a person acquires the first virus, sometimes you're asymptomatic. It's a mild case. The second time, however, based on something called antibody-dependent enhancement, people can have a hemorrhagic form. So you can get this virus 4 times. You can see from the pie chart here that 25% of the cases only are symptomatic. So 75% of people may have dengue, but may not have symptoms. It makes it very difficult epidemiologically to track these cases when you have 3/4 of the population not going in for testing, not showing that they actually might have the disease. Chikungunya is another virus we saw emerge in 2014. It comes from the Makonde language, which is a language spoken in Tanzania. That means the disease that makes you bend up in pain. Not one that I think anyone is interested in. It's characterized by severe arthritis, and this arthritis can persist up to 2 years after the infection. What's interesting about this one is you can see that 70% of the cases are symptomatic, and so it's much easier to keep track of where this disease is being transmitted. And then finally, Zika. Zika as you know, emerged in Brazil in 2015 and has strong links to congenital Zika syndrome. One of the characteristics of this is microcephaly. And you can see that, like dengue, we have so many cases that are asymptomatic that it's often very difficult to track. One of the things that's really interesting about Zika is that it's typically transmitted by the bite from an Aedes mosquito. It can be transmitted through blood transfusions, but it also can be transmitted through sexual contact. And so a person could be asymptomatic and still transmit this virus sexually. So it's very, very difficult to track. Most of the cases are from the bite from an infected mosquito, however. So those were the viruses. Let's talk a little bit about the culprit, the mosquito Aedes aegypti. This is a mosquito that's highly adaptable. It's typically a tropical or subtropical mosquito, but I did my dissertation research in the Sonoran Desert because this mosquito has adapted to an arid climate like the desert as well. It's what we call a human commensal organism, meaning that it preferentially feeds on humans. And so because of that, it lives near humans. This is not a mosquito that you find in swamps or wetlands or anything like that. No, this is a mosquito that you find in small or artificial containers, or large containers, artificial containers, in and around your house. This mosquito does not lay her eggs in the water. She lays it on the vertical walls of a container. So any container that has any amount of water and a wall is suitable breeding habitat for this mosquito. And that includes refrigerator pans, vases with cut flowers in your house, anything like that can provide habitat for this mosquito. She's also a day biter. So this may not seem that important here, but in the developing world, people often sleep under bed nets to protect themselves from mosquitoes. It doesn't work with this mosquito because she preferentially bites the first 3 hours after sunrise and the last 3 hours before sunset. So it's very difficult to avoid bites from this mosquito when you're out and about. So she transmits not only dengue, chikungunya, and Zika, but also yellow fever virus, a virus that we're seeing now in Brazil in huge numbers. Last year we saw a big outbreak in Angola and Democratic Republic of Congo. So this is something that sounds like it's from centuries ago, but we're still seeing yellow fever being transmitted. And then the last one here is the Mayoro virus, a new virus that we've seen recently in Haiti. So next I'd like to show you a video that will give you a little bit more information about the developmental stages of this mosquito. I'm not sure you've seen anything quite like this before. This is from a colleague of mine from the University of Arizona.
Once a female mosquito finds her host, possibly you, she begins probing to find a blood vessel. Female mosquitoes consume blood to acquire nutrients needed to produce eggs. In a single bite, she can ingest her own body weight in blood. While blood feeding, she is continuously injecting saliva, which can harbor deadly pathogens. Male mosquitoes are simply nectar feeders, and thus cannot transmit disease. Over the next 48 hours, the female mosquito will digest the blood and produce up to 100 eggs, which she lays in water-filled containers in your backyard. The eggs develop over the next 48 hours, after which they dry out and wait for the upcoming rains. Once wet, the larvae emerge within 45 minutes. Here you can see a first instar larvae struggling to emerge from the egg. The larvae mature over the next week to 10 days, depending on the temperature. They go through 4 larval molts, getting bigger at each step. During this stage, the mosquitoes filter feed while suspended upside down from the water's surface. You can observe the mouthparts beating as the larvae feed on bacteria and other particles. The larvae then enter the pupal stage and metamorphose into the adult mosquito over the next 2 days. Once the adult mosquito is fully formed, she splits open the pupal casing and emerges over the next 2 to 3 minutes, as you can see in this video, sped up 4 times normal speed. The newly emerged female mosquito mates and locates a new host, completing the life cycle.
Okay, so we've talked a little bit about the viruses, a little bit about the mosquito. What does it take for these viruses, these diseases to emerge? Well, you have to have a mosquito, Aedes aegypti, as you see here. You have to have the virus, and you have to have a susceptible human population, because once people have acquired these viruses, oftentimes they're immune. So you need a population that has not been previously exposed to the virus. Each of these elements has to be able to survive on its own, but within an environment that is conducive to the individual survival and to all three of them working together. And for this environment part, Andy Monaghan is going to talk a little bit about the weather and climate components that allow for this mosquito and its viruses to survive.
All right, Mary. First off, I want to ask a question, and there's probably some scientists here in the audience that would be able to answer this, but what is a model? Anyone? A model?
An abstraction from a population set on a given subject.
I think that that is a reasonable definition of a model. Any others? Okay. One way to think of a model is a representation of reality. And so in science, we often use mathematical representations of reality. And models that are developed here at NCAR, for example, can simulate weather and can simulate climate variability and predict what climate change might look like in the future. Those are mathematical representations of the climate system or meteorological interactions taking place. And we can also simulate these mosquitoes and simulate the viruses that they transmit. So why would we need models to study mosquitoes and viruses? Well, we would love to be able to observe these mosquitoes everywhere where they exist and occur on the globe, and see how their populations change in time, and look at the viruses they transmit, and observe all the cases of diseases that occur. But we can't do that. It's too big of a task, and it's too expensive to do. And so models are often a very low-cost way for us to observe systems that we can't fully, for us to evaluate systems that we can't fully observe. So what types of questions can these models help us answer? And today, I'm going to specifically talk about mosquito modeling. We typically address three types of questions with with mosquito models. Basic research questions are the first type, and those are really trying to understand just asking general fundamental questions about things that we don't fully understand, and they might help us to develop hypotheses and then go out into the field and test them. A second type of question that we address with models is related to decision support. And so we may be able to predict what the populations of mosquitoes will be a couple of weeks from now, and that can be informative for mosquito control districts that are trying to go out and do and coordinate their spraying activities. Or we can maybe do a seasonal forecast of whether or not mosquito populations might rise in a given area, and whether risk for a given virus would be higher than normal. And that can be informative for public health agencies. So kind of decision support purposes are the second types of question that we often will address with these models. And then the third type, which we increasingly get, is how are these viruses going to, or how are these mosquito populations, both their range and their seasonality, going to change in the future? And also the viruses that they transmit. So those are kind of the three categories that we look at. So you might ask, well, why are we running these models at NCAR? And that's a good question. The weather has a huge influence, it turns out, on vector-borne diseases and the vectors themselves. And by vector, I mean some sort of organism that can transmit a pathogen. In this case, this mosquito Aedes aegypti that Mary talked about earlier. Temperature, for example, affects the rate of development, the survival rates of mosquitoes, how large they grow, the rate at which they feed, and it even affects the incubation of viruses when the females take a blood meal. So here you have a picture of a female who's recently fed, and it can take a virus, depending on the virus and the temperature, from a few days up to a couple of weeks to actually incubate within the mosquito so that it can become a competent transmitter. So it can't just ingest a blood meal and then right away transmit the virus. And so temperature affects that rate as well. So it's a very important aspect. As the video Mary showed, pointed out, there are several aquatic stages for Aedes aegypti, and so we need rainfall for to fill up the containers that the eggs hatch in and that the larval and pupal stages occur in. And so we typically see that populations of mosquitoes go up during rainy seasons. And we also need humid enough conditions for survival. Mosquitoes typically like more humid areas, but they can often find little humid microclimates even within arid areas like Phoenix, for example. So what does a model look like, a mosquito model specifically? So here is an example of a dynamical model that we work with a collaborator at the University of Washington. By dynamical model, I mean a model that has a time step to it. It's kind of predicting at hourly or daily time steps. It's solving equations that describe the mosquito life cycle, and we can watch the life cycle evolve through time and watch populations of mosquitoes build up and shrink back down when it gets cooler. And you can see that the drivers of this model shown at the top of the diagram are temperature and precipitation and also daylight. And then we have equations that simulate the development and survival of each life stage. And then evaporation occurs in there, and that affects the amount of water that we have. It balances out with rainfall. And then land cover is also important. These mosquitoes are really adept at finding shady areas or sheltered areas and otherwise inhospitable environments. Okay, so there was a model. What do results from a model look like? These are the results of it's a daily animation through the year 2015 of adult Aedes aegypti populations in the United States. And you really think of this as suitability for the mosquitoes. It doesn't mean they're actually in these areas, but this model is simulating climatic suitability. And so the blue areas are a little bit less suitable, and the red areas are a little bit more suitable. And you can see here, we're in the month of May, and we have some in Florida, and then as we get into June, they start moving up, July, August, and then when we get to kind of the end of summer, we really have peak suitability for Aedes aegypti mosquitoes in the U.S. And then you can see as we get towards the end of September and into November, they start shrinking back down. And so you can see that models can be really useful tools for us to look at the seasonality of these, and that helps us understand the seasonal risk for virus transmission of dengue and chikungunya and Zika as well. Okay, so I wanted to talk specifically about a model that we've been developing here called WATCHM, and it stands for Water Height and Temperature in Container Habitats Energy Model, which is a mouthful. And we developed this model, so we do quite a bit of field work, and we've noticed just a huge diversity of containers that we find in people's backyards that mosquitoes breed in. And there are certain ones that they seem to prefer and others that they don't. And so we set out to develop a model that could help us investigate what the temperature and water volume are, the water temperature and volume of water are in each of these containers, depending on the container characteristics and the local meteorology. And that's what this model does. So what it does, it's called an energy balance model. And I don't put the equation up here because I want you to memorize it, I put it up here because I want you to keep in mind again that we're talking about models, and really what a model is a mathematical representation of reality. And so this is an energy balance model here that we're talking about, and each of these terms has a more mathematical representation of it that we solve for. But what an energy balance model is basically doing in this case is solving for the energy inputs to a water-filled container, and then the exchanges of energy that it has with the surrounding environment, with the surrounding air and the ground and whatnot. And then the residual of that equation, or the end result of that, is the solution for the temperature of the water and the volume of water in the container. So that can be really helpful for us. We can then plug this model into that mosquito model that I just showed you, and it helps us to more realistically simulate the populations of mosquitoes we have, and in particular to more realistically simulate the containers that might be in a given area locally. So like any model, we can't just develop and use it. We need to go out and evaluate it or validate it. We need to compare it to some observations. And so for this model, in 2013, after we developed this model, we went down to Veracruz, Mexico, which is an area where we've been working for a number of years. And it's a perfect spot for mosquitoes. It's on the coast in central Mexico. It's humid, it's warm, tons of Aedes aegypti there. And you can see in this photo on the top that there are 4 different containers. There's a gray garbage can you can see near the top, and then a white 5-gallon bucket, a black 5-gallon bucket, and then a black 2-gallon bucket. And these are typical of the containers that we would find in backyards, amongst other ones as well. And so what we did then is place temperature sensors, which you can see in this photo here. We suspended them in each of these types of containers, and each of them is water filled. And then we followed the temperatures. We recorded the temperatures hourly over a 1-month period. And these containers here are in full sunlight. What you can't see in this photo is that there are another 4 identical containers on the same premise about 20, 25 feet away. And those are in partial sunlight. So they're kind of partially shaded. They're getting sunlight part of the day, but not all of the day. And then also about 25 feet away, we have another set of 4 identical containers, and those are in full shade. So they're not getting any direct sunlight, they're only getting kind of diffuse sunlight. So we have 3 sets of these 4 containers, and we're monitoring them all over a 1-month period. And here are some results. So I'm circling this black bucket here because these are the results from this very bucket. And this is, again, a bucket in full sunlight. And on the vertical axis, we have temperature. And on the horizontal axis, we have the day over this 1-month period. So we're looking at the temperature fluctuations over 1 month. And in black, we have the observed temperature fluctuations. And in red, we have the watch-em, the simulated container temperatures from our model. And then this little purple bar that we put up here, I put this up here because this is kind of the sweet spot for development and survival of these mosquitoes. It goes from about 25 to 32 degrees Celsius. That's about 75 to 90 degrees Fahrenheit. It's really, if they could have any sort of temperature range, this is where they would want to be. And so you can see from this that it's not a great situation for them. They're in that ideal zone for part of the time, but it gets really hot during the day here in the this direct sunlight. And so this might not be an ideal container for these mosquitoes to breed in. But if we go about 25 feet away and look at the exact same black bucket that's fully shaded, these on the bottom here are the observed temperatures that we see and the modeled temperatures that we see. And you can see that in this case, the temperatures hardly ever go out of that sweet spot. So you would expect that if you were going to find mosquitoes, you would probably be looking in that shaded bucket. And in fact, when we're in the field, this is something that we usually see. We see that containers that are well shaded are ones where we typically find immature mosquitoes breeding. And so Watch'em, as you can see, does a pretty good job of predicting what we observed in those buckets. So it tells us that our model is doing a pretty good job, giving us reasonable results. And also that there are huge temperature differences amongst these 2 buckets that are on the same site. So just the type of bucket that you have and the conditions that it's in are really important for being able to accurately simulate mosquito populations then. So we can then take and put this model back into that larger mosquito life cycle model that I showed you and do a better job of simulating those. We can also use it to answer some kind of really weird hypothetical questions, and this is one of those. Here we have, we just simulated over a 1-year period, what would happen if we put a plastic barrel at every spot in the United States in partial shade. So there's a partially shaded kind of a 50-gallon, 55-gallon drum everywhere across the U.S., and we simulated the water temperature and volume in it with Watcham. How many generations of larvae per year could we put through there? So this means how many times could a larvae go through its larval cycle in 1 year? And so the red areas are where we could actually get about 52 cycles, so almost one a week on average over the course of the year. And the blue areas are where we have, maybe 10 to 20 cycles a year, so far fewer. And so you'd expect the red areas to be where we'd have much more suitability for these mosquitoes. And then on the bottom, we have the exact same map, but it's for a soda bottle that's in the shade, and we find these quite a bit in backyards. And what we see here, if you just kind of glance and compare these two maps, is that overall it's much more suitable in this plastic bottle. You can see, for example, if you look at South Florida, it's red for the plastic bottle. We can get around 50 cycles a year, whereas in South Florida on the top, we can get maybe 30 cycles a year. An interesting thing about this map is that if you put these two same containers in full sunlight instead of partial shade, the results would probably flip. That pop bottle, because it's such a small volume of water in these more southerly areas, is going to get too hot during the day in the full sun, just like I showed you in the previous slide, whereas this bigger volume of water that's in the 55-gallon drum, it doesn't have the big swings in temperature that we see in the smaller bottle. So the result here depends also on whether or not this is a shaded container. So I just want to summarize where we're at here. This is the same animation I showed you earlier, but just again summarizing that we can use models like Watcham to improve our simulations of mosquito life cycles. Okay, so I'm just about done here, but I wanted to just stop and tell you that there are simpler ways to look to model different aspects of mosquitoes that we want to understand as well. So that was a dynamic model, and as you can tell, it's pretty complex. There's a lot going on, but sometimes we just need simple answers, and we need them quickly, and we don't want to spend a lot of computational time on them, and so we can develop empirical models as well. And empirical models are more of a statistical representation of reality. So the dynamical models, we're actually trying to use mathematics to solve for different aspects of the life cycle of the mosquito. These ones are more of a black box. We have some observations around the world, not everywhere, but we have them in a handful of locations of where we find this mosquito, and we know what the climatic conditions there. And we can basically develop a correlative model or an empirical model of just a real simple equation that says, okay, if the temperatures are in this range and the precipitation's in this range, this is where you'd expect to find this mosquito. And so that's what we're doing here. This is a paper that we published last year looking at different suitability areas for this mosquito. It's simply based on temperature and precipitation thresholds. It's what we call a fuzzy logic model. But anyways, the point here is that we don't know, we haven't been able to observe everywhere where this mosquito probably lives on the planet. And so maps like this can help us. We do have climate information everywhere on the planet. So if we can develop a model like this at the few points where we do have both mosquito data and climate data, then we can extrapolate where it might be suitable for these mosquitoes using the climate data. And so that's what these types of maps help us to get, and they're really fast and easy for us to develop. And they can be useful for a lot of things. This is the same model, and here we're using it for a climate change application. And so just to orient you with this map, the lighter colored shading on the bottom, which is kind of mauvish, is the present day range of this mosquito in the US. So by the way, we do not have Aedes aegypti in Colorado. So we don't, and we don't have its cousin Aedes albopictus either. So we don't have risk for local transmission of these viruses by mosquitoes in Colorado. But anyways, as you can see, we have this mosquito across the southern tier of the U.S. It's been introduced in California since 2013, so it's relatively new there. And if we take that model that's based on temperature and precipitation conditions, and then we use climate models developed here at NCAR to predict what might happen 50 years from now, this is the red map that we see, the red areas we see here. So in addition to the mauve areas, 50 years from now, around 2070, we might expect that the range will be suitable for this mosquito in these further north areas that are in dark red. But there's something interesting about this map here. If you look towards the bottom in Florida and Texas, there's a bunch of blue stars. Those are the areas that Mary discussed briefly earlier where we have had transmission of these viruses locally in the United States. So you can see that really, even though we have the mosquito across a broad swath of the South, we really see virus transmission in these very far southerly areas. And so that tells us something about needing to have a long enough season for these mosquitoes to have virus transmission. And what we find is that areas where we have year-round or near year-round suitability for the mosquitoes, where they could basically live year-round or nearly, those are the areas where we see the highest risk for virus transmission, and those happen to be South Florida and South Texas. And so we can take the same model and look at where it predicts areas of year-round or near year-round mosquito suitability in the U.S., and that's what we're doing here. And these are areas where we might expect the virus transmission risk for Zika and chikungunya and dengue to be the highest, both now and if you go into the dark reds into the future. So you can see 50 years from now, Atlanta might be a much higher risk area, for example. Okay, so I'm done speaking about models, and I'm going to pass it back off to Mary, because one of the things that we really want to drive home is that even though we can model many of the weather factors that affect mosquitoes and the viruses they transmit, these models can only go so far. As you can imagine, human behavior, socioeconomic conditions, and many other human factors have profound impacts on risk for having mosquitoes and virus transmission. And so that's really at the core of what Mary does. And so I'm going to pass it back off to her to talk about that.
Great, thanks, Andy. So I'm going to talk a little bit about the work that we do here to help us better understand the drivers of risk for these viruses. And one of the ways we do this is by assessing the local human risk. Here we're talking about dengue virus. So you saw this diagram earlier. I'm going to talk a little bit about Aedes aegypti. I'm going to talk about the humans. And then DENV is how we denote dengue virus, not Denver. But I'm going to talk about, from the map Andy showed, you saw that, and he talked about South Florida being an area where we've seen transmission, but he also showed that there are mosquito populations in Arizona, and I mentioned that earlier, that I had done my dissertation work there. So we're interested in looking at the differences between Key West, Florida, and Tucson, Arizona, for example. We know one area has had transmission and the other doesn't, and yet both have mosquitoes. So our goals with this study were to better understand the drivers of dengue transmission, but also to inform decision makers about prevention and response. And so the work that we're doing is not just an academic exercise. We're interested in working with our stakeholders and decision makers at all times to ensure that our research is relevant to them. So I'm going to show you a case study of these two different cities, one with transmission and one without. And one of the questions we're asking ourselves is, are there different risk factors in these two places? So here on the left, I have the factors, then I have Tucson and Key West, Florida. So is there the potential for viral introduction? Well, in Tucson, I worked in the Nogales, Ambos Nogales area, which is right on the US-Mexico border. About 13,000 people a day cross this border. So there's constant potential for viral introduction. And in Key West, Florida, 2 to 3 times a week, cruise ships pull in from all around the Caribbean. So there's also the potential for viral introduction in both of these areas. Is there a susceptible human population? Well, Tucson, we haven't had transmission, so we assume that people are immunologically naive. Key West, we know there has been transmission, so people are susceptible. How about established Aedes aegypti population? Well, we know for a fact since 1994 in Tucson, and I mentioned that centuries and years ago, we've had Aedes aegypti around the country, but in recent years, 1994, we saw the first documented reintroduction of Aedes aegypti, and Key West, I think, has always had Aedes aegypti since the 1700s. Is the environment conducive for vector-human contact? So this is one that we're not really sure what's going on in Tucson. Are people out at the times of day when the mosquito's biting? We know that in Key West, the lifestyle is such that people sit outside all the time. It's a very tropical environment, and people are very interested in outdoor activities. Tucson, we weren't so sure. Okay, and then is the environment conducive for viral development? Andy mentioned to you that for the virus to circulate within the mosquito, it takes a certain amount of time. It can take from 3 days to 12 days or even longer. It's temperature dependent, but it also may be dependent on other factors. And so these are the kinds of questions that we're asking ourselves. We know in Key West that definitely it's conducive because we've had transmission, but we don't know what's happening in Tucson. So we know that Key West, as I mentioned, is a tropical, subtropical environment. On the right, you can see an example of one of our households. And Tucson is a very arid environment, very dry in the Sonoran Desert. So totally different environmental settings, but two in which the mosquito is around. So what are some of the questions that we ask to help us better understand some of these human factors? What we've done here is in each of these cities, we've conducted a 400 household survey. So we've gone door to door asking people. These are randomly selected households. And we have a certain set of questions that we try to get at to give us a better understanding of what's going on from the human side. The first one is knowledge. We want to know if people know how these viruses are transmitted. Are they even aware of the fact that they're transmitted by mosquitoes? Or in the case of Zika, do they know about sexual transmission? We want to know if people know where these mosquitoes lay their eggs. And this is not a very simple question because, as I mentioned to you, they will lay their eggs in anything that has water in it, any container that has a vertical wall and water. That means a bottle cap. That would work too. We've seen mosquito larvae and pupae in folds of tarps, all kinds of things. So anything that can contain water, and people aren't usually aware of that. And then what type of mosquito prevention strategies are they undertaking or are available for them? We're interested in their attitudes. Do they think dengue is a problem? Because if they don't, they're not going to take any action to reduce their risk. And what about the mosquito burden? Do they notice a lot of mosquitoes around? Do they think that they're getting bitten? This is something that we know from a study we did in 2003 here in Colorado, that we had so much West Nile transmission here, and everybody we asked said, oh, we don't have mosquitoes here. They don't bother us at all. 80 to 100,000 people were infected in 2003 with West Nile along the Front Range. And then willingness to pay for mosquito control. Are people interested enough that they are willing to put some more money into controlling mosquitoes? Practices. So what do people do to reduce mosquitoes? Do they actually pay attention to public health messages, which are asking them to empty standing water from around their households? Do they do that? Do they check their gutters to make sure there's no standing water there or debris that the mosquitoes could survive on? And what do they do to avoid mosquitoes? Do they actually pay attention to public health messages about wearing long sleeves and long pants, wearing repellent, staying indoors during the peak biting times? And do they seek health care if they have a fever? Because this is one of the big problems, right? We don't know how diseases are being transmitted if people don't go to the doctor and don't get tested. And so we were interested in these types of questions in both cities. So the first question is, why is there no dengue in Tucson? And is it because of human behavior? So again, we have the factors, then we have Tucson, and we have Key West. And so when we asked about using insect repellent, we can see that it's pretty much the same in both places. Half the population never uses insect repellent. Restricting time outdoors, wearing protective clothing, using screens and or air conditioning. These are all pretty much the same in both. There's no statistical significance to these. But when we ask other questions, so in Tucson, 43% of the population was not aware of dengue, compared to 23% in Key West. 29% of the population in Tucson never removed their standing water, compared to 19% in Key West. And then 21% traveled to dengue endemic countries in the past 12 months in Tucson versus 15% in Key West, Florida. So by all accounts, it looks like Tucson is at higher risk based on the human behavioral component. Well, so maybe that's not it. Maybe it's the mosquitoes. So why is there no dengue in Tucson when there are more Aedes aegypti? Well, we have the factors, again, Tucson and then Key West. In Tucson, a bit fewer people notice mosquitoes than in Key West, Florida, but again, not statistically significant. But when we look at the number of containers with water per house, we see that there are more in Tucson than in Key West, Florida. And when we look at the number of households with at least one Aedes aegypti pupa, we see that there are more in Tucson versus Key West. And I want to just explain what we do with this. And so we go to people and we ask them, do you empty your standing water from around your house? Do you take precautions against mosquitoes breeding in your backyard? Oh yes, we do. So we actually go out and look. We collect the mosquitoes from their backyard. We count the number of containers they have. We count the number of pupae, so we can actually see if what they say is really holding to what we find in their backyards. So you can see that mosquitoes are also not the cause here. Excuse me. And so what that leads us to a couple of conclusions. One, we find that Tucson would seemingly be at higher risk based on these data. It's possible that there are epidemics taking place here and we just don't know them. Remember I said 75% were asymptomatic, but highly unlikely. We would probably know it if that were the case. And so another conclusion that we've arrived at is that it's possible that the humidity levels are so low in Tucson that the mosquito is not living long enough, for this virus to cycle. This is one of the hypotheses that we're actually testing at this moment. We know that it takes 8 to 12 days for the mosquito to have this virus cycle within her. And in Tucson, we're finding that the mosquitoes really don't live much longer than 10 to 12 days. So we think that that's one of the reasons. So these are the types of studies that we do, but we're also really interested in what we can do to intervene. What is it that we can do to move beyond, as I said, just collecting academic data, looking at risk factors, but actually doing something that will help us work with the public to reduce mosquito populations? So one of the questions that we asked on our survey in Key West was something that was of interest to the vector control units there, one of our stakeholders in this study. And it had to do with genetically modified sterile mosquitoes. So these are male mosquitoes that have been sterilized. They are then released and mate with wild females. And because they're sterile, there are no progeny. So we see a reduction in the number of mosquitoes. This has been a very hot button topic in Key West. For the last 5 years, they've been talking about this. So in our survey, we find that 58% of the population was supportive of the release of these mosquitoes. 28% of the population was neutral. And when we looked at some of our qualitative data, because we always ask people, well, why are you supportive? Or why are you neutral? Why are you saying that? We find that people were neutral because they felt like they didn't have enough information. They wanted more information. So we were able to go back to our stakeholder and say, you need to hold more town hall meetings. You need to get this information out to the public. 14% were opposed. And I've told this story to Andy before, but we did randomly selected households in Key West, 400 of them. I happened to draw randomly the guy who was leading the opposition to the release of these genetically modified sterile mosquitoes. 2 hours I spent at his house while he talked to me about this. And what happened was they put this to a vote again at the Florida Keys, Monroe County, and they actually approved it last year in November. So we'll see what happens. The release has not taken place yet, but it's something that is of great interest to us. So that's one option for how to reduce the number of mosquitoes. Another thing we're working on is what we call scalable solutions. So working at in other areas at a very, very local area, working with the communities. And one of the options that we have is to use an app, which is a climate-driven mosquito alert app. And so why are we doing this? Well, obviously, from what I've told you, we need to reduce the number of these re-emerging and emerging diseases in our country. And so we're trying to use this app as an early warning system because we think that if people have this app and they see that conditions are ripe for heightened mosquito or more mosquitoes, mosquito abundance, that this will trigger them to reduce the standing water in their homes and reduce the number of mosquitoes. And so our goal is to develop shareable information that can be shared across communities. And one of the things that we do here is push out these messages that say something like, your area has experienced rainfall in the last 4 days. Based on temperature conditions, mosquito eggs can develop into adults within 6 days. Empty standing water in and around your house. So we're still working on this, and we're hoping to bring it into a project that we're starting, or we hope to start. And so I want to get your input on some of these things. So I want to ask you now to vote on a couple of different faces for this app, and let me know which one you think might be more effective. Okay, ready? Here's A. Okay, that's A again. Here's B. And C. Okay, so I'm going to ask you to actually vote, and Larissa's going to help me with this, right? Keeping a track of it. How many of you like A? Ooh, not too many on A, okay. How about B? Okay, how about C? Ooh, close. All right. Okay, so it's pretty close between B and C. A's out. All right. So I'd like to thank you for that, because this is something that we'll definitely utilize when we get this app going. And I'd like to now just leave you with a public health message. I can't help myself from doing this, so how can you reduce your risk at home and abroad? First of all, you need to be aware of vector-borne disease risks. If you're traveling to India or Nicaragua or Guatemala or Brownsville or Key West, you need to be aware of any public health messages that are out there about mosquito-borne viruses that are being transmitted. At home, you also can empty your containers with standing water. The Culex mosquitoes that transmit West Nile in Colorado are also container breeders. It's important that you have no standing water around your home. If you can't empty something, and I have drains at my house that I can't empty, and so I put in this bacillus dunk that you can get at Home Depot. It will kill only mosquito larvae. It won't kill anything else. It won't kill fly larvae, just mosquito larvae. So it's perfectly safe for animals, birds, humans. It's only detrimental to mosquito larvae. So I highly recommend doing that if you can't empty your water.
What is it?
It's a bacillus. It's called BTI, Bacillus thuringiensis israeliensis, and you can get it in little dunks. They come in a 6-pack at Home Depot, and I'm not doing an ad for Home Depot. I'm sure you could do it at Lowe's too, or some other place that has these.
Any hardware store.
There we go. Any hardware store, yes, would have it. And then make sure that your windows and doors have screens that are completely repaired. When I ask this question, and I ask this all the time when I'm doing surveys, are all your window screens and door screens intact? People always say yes, as the cat jumps through the door screen. Yes, no matter what you ask them, they say yes. Oh yes, everything's fine. So check that, because mosquitoes are notorious for finding any little hole. If you're going abroad, staying in an air-conditioned room, believe me, I travel a lot in a lot of places where mosquito-borne diseases are being transmitted, and I always try to find air conditioning. It doesn't completely prevent mosquitoes from coming in, but it definitely reduces it. They don't like cold temperatures, as you saw from Andy's models. And you need to take the appropriate medication when traveling. I hear this all the time. People, oh, I'm only going to Ghana for 2 days. I don't need malaria meds. Oh yeah, you do, because they'll find you. So, home and abroad, avoid mosquito bites. You need to wear long sleeve shirts and long pants. And remember, Aedes aegypti bites during the day. The Culex mosquitoes that transmit West Nile, the Anopheline mosquitoes that transmit malaria, they bite from dusk to dawn. So sometimes When I'm in places like Ghana, for example, I'm wearing repellent day and night because I don't want to get bitten. And you have to avoid mosquito bites by using repellent or insecticides in and around your home. And I recommend that you look at the CDC or EPA websites to see what they're recommending for safe repellents. So I'd like to last put up this acknowledgement page. As you can see, this is really, it takes a village to do this. This is not just something that Andy and I can do alone. We do a lot of field work, and we have collaborators from all over. It's your cue to come up.
Okay.
So Mary talked a little bit earlier about, she showed a chart showing the ingredients for virus transmission for Zika or chikungunya or dengue, and it had human factors. You need to have humans, and they need to be exposed to the mosquito. You need to have the virus introduced, usually through travel to get it into the U.S. And you need to have the mosquito populations, and as you saw, we can simulate those with models. And you need to have suitable environmental and climatic conditions. So we, as you can tell, our program is aimed at addressing all of these ingredients that you need, not just the meteorological ones. And so last year, about this time, we wrote a paper where we put together all these factors. We simulated mosquitoes. We looked at travelers coming in from Zika affected areas. We looked at indicators of human exposure, outdoor exposure to the mosquito, and some other factors. We put those together into and made a series of risk maps that we published. And it turns out that they were really heavily used, and that was really gratifying for us, I think. I think as a scientist, you always want to strive to have some societal impact for your work. And this here is an image of President Obama's press secretary using one of our maps. You can see it in the background. And this is when Congress, the White House was making the case to Congress to fund a response to the Zika pandemic. And that eventually was funded, and the funds are being used for development of a vaccine and to beef up some of the mosquito control activities we have in the U.S. and to deal with the outbreak that's been biggest in Puerto Rico. So anyways, I think that's all we have to say, and we'd be happy to take your questions.
So